Hi, welcome to Ibrahim Academia. Today we are going to discuss the mechanism of hepatitis B virus, that how hepatitis B virus enters a human body and how it fixes with the NTCP receptor upon the hepatocytes, how it gets inside the hepatocyte cell, and then how it causes the infection, how it replicates inside the hepatocytes, and after that how my immune cells recognize this infection. All this will be discussed in this lecture. Let me first tell you that hepatitis B virus belongs to a family which is called Tenofoviridae. Before this, we discussed hepatitis C virus, which belongs to Flaviviridae family. This hepatitis B virus comprises of a lipid bilayer, the outermost layer. This layer is not the part of the hepatitis B virus. It is my part. This layer has been stolen from my cell, from my endoplasmic reticulum or my Golgi apparatus. After that, there is capsid protein. This capsid protein is called HBCAG, hepatitis B virus capsid antigen, and this is called hepatitis B virus surface antigen. This is surface, and these are surface antigens, and this black is HBCAG. This surface antigens has a very much big role for the attachment with the hepatocytes. And whenever this hepatitis B virus enters someone's blood, this hepatitis B virus is in search of a gate, is in search of a receptor which is called NTCP. This receptor is present on the hepatocytes. It is called sodium taurocholate co-transporting polypeptide. This hepatitis B virus HBsAg part, the long part, is fixed with the NTCP receptor and it is endocytosed inside the hepatocytes. Whenever this hepatitis B virus endocytosed inside the hepatocytes, it finds ways to enter into the nucleus of the host. This capsid molecule gets entry into the nucleus and the entire genome has been released inside the nucleus. Okay, this rcDNA entered inside the hepatocyte cell inside the nucleus. This rcDNA is converted into cccDNA, covalently closed circular DNA. Who convert this rcDNA into ccC? This has been converted by my own polymerase enzyme, by my own repair enzymes. After the cccDNA, there will be transcription of multiple RNA molecules, and for that there is RNA polymerase 2, which is my own enzyme. This will come and transcribe the RNA molecules, messenger RNA plus the pgRNA. This is very much important RNA molecule that could be used for the future purposes for the new virions. These messenger RNAs and pgRNA comes out of the nucleus of the host cell, and on ribosome they gets the translation by my own ribosomes, and the all the antigens, the HBs, HBC and HBX, all these has been formed by translation of my own ribosomes from these messenger RNAs. What does the pgRNA do? pgRNA acts as a template for the new virus particles. The polymerase enzyme, it forms the polymerase enzyme, it forms the HBCAG protein, and when the polymerase enzyme, this enzyme form, this starts the reverse transcription of this pgRNA and form the negative strand DNA molecule. And when the negative strand DNA molecule form, it then form the positive DNA strand molecule, and when it reaches half of the DNA negative strand, then it stops suddenly and the entire capsid gets its assembly and new virus particle has been formed, and those new viral particles starts to infect the new healthy viruses and also with the immune response. In this case, some molecules were excreted by the infected hepatocytes. These are interferons and interleukins. These are the immune response proteins, immune response signals, and along with that the Kupffer cells inside the liver. These cells also start the representation of a special molecule, a special protein which is called HBsAg. This molecule is presented to the T cells, especially the CD8 T cells, which are cytotoxic. These cytotoxic T cells start the killing of these infected T cells. But in some cases, when the hepatitis B virus dominates over the activity of the CD8 T cells, then the infection may find its way towards chronic form. And this chronicity is just because of the cccDNA residing inside the nucleus of the host, and the case may go towards the fibrosis, then the cirrhosis, and then carcinoma. Now, for the treatment of hepatitis B viruses, all the drugs contains nucleosides. They are nucleoside providers. Those nucleosides, when get incorporated inside the DNA during the reverse transcription by the polymerase enzyme of the hepatitis B virus, so the entire process of replication stops, and there is difficulty for the virus to replicate. These drugs have this kind of mechanism, but along with the drug there is a special kind of vaccine which is called recombinant HBsAg vaccine, recombinant hepatitis B virus surface antigen vaccines. In this case, those antigens, the genes responsible for this kind of antigen has been cut from that virus, and it is incorporated inside the E. coli, inside other bacteria, inside the yeast and the laboratory. And that gene, after proliferation, after transcription, translation, the protein of that gene, which is called HBsAg, has been synthesized, and that protein can now act as a vaccine for the babies. Those vaccines, when administered in babies, so the macrophages present that vaccine to the T cell and B cell. The B cells and T cells form memory cells and antibodies, and those antibodies are very much special against the agglutination of these kind of antigens. After that childhood period, whenever there is an entry of HBV virus, so the HBV virus already has surface HBsAg antigen. That antigen can be detected by the memory cells, and the antibodies of the memory cells, they agglutinize this part, this area, and the virus gets stuck, and those viruses can't enter inside the hepatocytes, and the infection stops at early stages. This is the mechanism of HBs vaccines. Thank you very much. Stay tuned to the Ibrahim Academia. Today we are going to discuss the uric acid problem in human.